So welcome everyone. My name is Lisa Horton. I'm part of the board of the Wood River chapter of the Idaho Native Plant Society. My co-host tonight is Mary McClanahan, who also is on the board of our chapter. Tonight, our presentation is The Fruits of Botanical Exploration with Dr. Don Mansfield. I'm going to turn this over to Mary McClanahan, who will introduce Dr. Mansfield.
Thanks, Lisa. I am so excited to have Dr. Mansfield give a presentation tonight. I first became aware of his work when he gave a presentation for the Idaho Native Plant Society and in September. And it's on the Idaho Native Plant Society YouTube channel. And it's just got me so excited. And then when I really got excited was I was trying to fill out a species list for a Wood River chapter of the Plant Society hike that we did in the Sawtooth. And I got on online and I saw that Dr. Mansfield had collected plants all over central Idaho. And that got me even more excited. And so I was very thrilled when I asked him, and he agreed to give a presentation tonight. And Don Mansfield got his doctorate degree at Idaho State in 1979. And I think he thought he was just going to be a teacher, a college teacher, a college professor, which would have been great. But it looks like his life kind of kept drawing him toward more specific botanical research. So he got an undergraduate degree in biology at Colorado College, and then he got a master's in botany in British Columbia. And then he got his doctorate at Idaho State. He kind of bounced around. It looks like Dr. Mansfield, you taught botany or taught at several liberal arts colleges in Florida and Colorado. But it's just so great that you ended up back in Idaho and at the College of Idaho, which is a liberal arts school in Caldwell. And it's a small school, but it's a very dynamic school. And Dr. Mansfield has really worked to upgrade the herbarium at the College of Idaho. It has 60,000 specimens. And I've worked with some of those specimens when I was doing botanical surveys across the Owyhees. And he also found that there were some collections that were misidentified. So he's helped clear those up. And then when he goes out and he's done a lot of his work in Southwest Idaho, Northern Nevada, and Southeast Oregon, and he goes out and he finds plants that no one ever knew was there. And so he's finding rare plants, and he's finding plants that are very common in central Idaho that no one knew was down there. And he's working especially hard on the biscuit root genus Lomatium. And he's also published a guide to the flora of Esteens Mountain in Eastern Oregon. So he's just a very exciting leader in botany in Idaho. And I'm so happy that he's giving our presentation tonight. And thank you so much for coming, Dr. Mansfield.
Oh, you're very welcome. Let's see, this is the one I want to share. I think we're good to go, aren't we? Everybody hear me okay? Lisa, good? Mary? Okay, great. All right, I'm going to talk to you about the fruits of botanical exploration. And to do that, I'm going to, oops, if I can advance the slide, that will help. I can't, oh, there we go. Oh boy, that's not going to be the way to do it. Okay, well, let's see what we can do here.
You can try your arrow key.
Yeah, that's what I tried. It's not doing anything. Okay, it's going to be tricky because I'm doing it with my mouse. But anyway, we'll see. I want to put a little historical perspective on it and talk about what floristic botanists do and the fruits of their work. And I'm a floristic botanist, and I'm going to talk about, first of all, sort of the history of floristics in Idaho, very briefly, give you some examples of my recent explorations and how my study of the distribution of plants in a particular area can give rise to discovery of new species, which is one of the really important ongoing aspects of botanical exploration. We're finding new things that we have never thought were here in ongoing species descriptions. We are also, I'm then going to talk about one of the resources that to which Mary was referring. And then finally, I'm going to come to central Idaho to talk about some things that are closer to your home, which are some of the central Idaho endemics and rare plants. And you'll see I'm drawing heavily on the slides of one member of the audience right now, Michael Mancuso, who kindly donated a whole lot of the slides I'm going to show you this evening. I'm using several of his here. So I want to start, and I don't know how to get rid of that stuff up the top there. Well, at any rate, the first plants discovered in Idaho, if you will, other than those used by indigenous peoples who lived here long before the Corps of Discovery came in 1805. But Lewis and Clark expedition came down the Clearwater River, and in September of 1805, collected the first specimens of ponderosa pine on the right of the, can you see my pointer here? Oh, good. Yes. Okay, great. Of Idaho fescue, of American bistort, of old man's whiskers. And they then had these collections that were then ultimately sent back. On their way back from the Fort Clatsop, where they wintered in 1805, 1806, the spring plants were out, and they collected more plants on their way back. And these were really the first collections of plants from that first exploration. The phlox up on the right, the Herbarium, what's the common name? Oh my goodness, it's escaping me. That's right, false hellebore. Thanks. Scarlet Gilia. And the one down on the bottom I'll come back to and talk a little bit more later. That's the 9-leaf biscuit root. And so those plants were collected, sent back, and then in the 19th century, there was quite a series of explorations, and those explorations gave rise to descriptions of many of Idaho's plants, mostly the common plants, like you see here, bitterbrush here, sagebrush, camas, and coyote willow. Now, camas was one of the first to be described shortly thereafter by Persh, because camas was widely used as a food source for the Shoshone and Nez Perce and a number of other of tribes. And so those plants, of course, either common ones or ones that were used by indigenous cultures, were the first to be described. And oftentimes these were sent back to botanists back east, collected by explorers who were hired as part of the expeditions that explored the West in the earlier days. By 1895, the explorations were starting to be conducted by people living in Idaho, and L.F. Henderson really conducted the first, to my knowledge, and I think the first, Rhoda Love at Oregon State has written a lot about these explorations. In 1895, L.F. Henderson left the young University of Idaho and set forth in the summer on a long expedition that took him down through Long Valley, down into the northern Snake River Plain, up the Wood River Valley to Stanley, down the Salmon River, and then down the Lost River, Big Lost River Valley. He missed the Little Lost and went up the Lemhi Valley and up to Salmon. And that was a rather lengthy collection, most of which, unfortunately, was lost because the herbarium at University of Idaho burned down shortly thereafter. But a number of other collectors continued, and I'm only going to mention briefly any of them because there were so many. And fast forward, oops, let me advance this. We will fast forward to the mid 20th century, by which time a number of explorations had been conducted, a number of herbaria had distributions of Idaho plants. And this was the first flora of Idaho. It's the only flora of Idaho to date. Ray Davis in 1952 at Idaho State University had assembled enough collections to do the first, what we call floristic volume, the first flora. You can see there, it's the flora of Idaho. And that describes all of the known species of Idaho at that time. And he was able to do that by having all these collections come into his herbarium. And he's then able to describe all the different species that had been named by other people. And a lot of this was he named, a lot of them were in the process of making the flora. You recognize that there were several what we call new combinations. And then during the same period, mid 20th century, Leo Hitchcock and Art Cronquist up at the Hitchcock out of the University of Washington. Cronquist, a student of Ray Davis, worked on this flora of the Pacific Northwest. Now, what you see here is a single volume that came out in 1973. And a lot of you can remember that time. That was actually a short version. It's about that thick, so it's not terribly short, but it was a multi-volume set, 6 volumes or 6 volumes, that this was a short version of. And until about 2 years ago, this was the go-to for Idaho plants north of the Snake River Plain. So any of you who've done any explorations in Idaho, looking at plants and trying to figure out what they are, this is the go-to book. Was until about 2 years ago. Explorations continued in the late 20th and early 21st century. I've just got a handful of the many people that have been involved in more recent explorations. Douglas Henderson out of University of Idaho was a prominent botanist in East Central Idaho and made many of the collections I'm going to show you later. And Bob Mosley and Mike Mancuso, who's here in the audience, also were students of Douglas Henderson's, and others, Steve and Pam Brunsfeld up at University of Idaho, Pat Packard at College of Idaho, Barbara Erder, who's a prominent floristic botanist, Curtis Bjork, who now lives in British Columbia, and I could go on and on. This is just a short list. Carol Prentice is another who's in the audience here. And as a result of these explorations, you heard me mention that north of the Snake River Plain was in the flora of the Pacific Northwest. But south of the Snake River Plain, that was covered by a different floristic treatment called the Intermountain Flora. Again, this is not a single volume. This is one of 6 volumes, and there is not a single volume that you can take into the field in the Intermountain region between the Rocky Mountains and the Sierras south of Rocky Mountains where you are. So the Snake River Plain down into Utah, northern Arizona, western Colorado, just a little bit of the eastern Sierra. When I came to the College of Idaho in 1989, my first project was to get a book that was a little bit more accessible to the student. And I wrote this book called The Flora of Steens Mountain by exploring an area just out of the picture here over in Harney County, Oregon. And wrote a flora. And in that flora, there are keys to the plants of that area, many of which hadn't been really recorded in a floristic treatment before. And as a result of my explorations, many subsequent species have been described in intervening years. And more recently, I've turned my attention to this region here. This is the drainage of the Owyhee River and the rivers and streams coming down off of the Owyhee Range over into the Bruno drainage. And so that's the area I've been most intensively collecting plants in and studying, because I am now I'm about halfway through a flora of southwestern Idaho, southeastern Oregon, and this bit of northern Nevada. Okay, let's see. So after my arrival at the College of Idaho, a lot of my work has been collaborative with students. And so we've been exploring collecting plants throughout the 90s, 2000s, and 2010s. And as a result of these explorations and our floristic study, when I say floristics, what I mean is the study of the diversity of all the plants in a particular area. And so the area I'm studying now is the Owyhee region. The area I first studied was Steens Mountain. Of course, my ongoing work and interest, as Mary pointed out, is Idaho in general, and I've done a lot of collecting throughout Idaho, but mostly in the Owyhee region. And so one of the major, what I'm going to do next is talk about a case study of the study of biscuit roots, or the genus Lomaceum, because as a result of floristic work, what you realize is that there are plants that don't quite fit in the floras that have been written. In other words, they don't read the books. And so I've been sleuthing the wild biscuit roots out of College of Idaho. Here's a biscuit root. This is a Lomaceum fruit here, shown here. So how do you recognize species? Because obviously, if you want to describe species, you want to know what you mean by a species. So I want to point out first, before we get much further, that this idea of a species is an abstraction. You can go out and you can look at a plant growing in a field or on a mountaintop or on a hillside, and you'll see others around it that are members of the same thing, and that's a population. And then you'll go to another mountain range and you might see a population of that same thing, species. It looks a little different. You might go somewhere else that sees a little bit different. Well, so how do you recognize that they're the same thing, the same species? And these are abstractions, and the labels, species, are our way of really organizing this diversity into manageable units. And these manageable units do make sense, not always, but usually. And it really is helpful to be able to know which ones are edible and which ones will kill you, which ones have medicine, which ones don't, which ones are rare and which are common, which can grow in my garden and which can't. So knowing the species really matters. So, whoops, better do it this way. Here we go. So how do you know what a species is if they're abstractions? Well, some species are really fairly easy. They read the books. And those have characters that they share in common. So for example, if you look at sunflowers, annual sunflowers, they all have certain characteristics in common that they share with other populations of sunflowers in other locations. And they're a little bit different than, say, Cusick sunflower, which has characteristics in common with other populations of Cusick sunflowers, but they're a little different than annual sunflowers. Now, why are they different species then? Well, because you can see them and recognize by looking at them that they're different. It's not always the case, but that's a good bet. Now, why do they look alike? Well, because they interbreed, and that's kind of a criterion for what makes a species different from another species. Similarly, they tend to be living in different areas. So ecologically, they're different. And that's another criterion for recognizing a species. Members of the same species will be populations that live in similar habitats, or at least habitats that species has adapted to. Another criterion is that all members of the same species really have a common ancestor. That is, they're genetically related. And so when they interbreed, we say they share a common gene pool. Where they get that gene pool? From their common ancestor. Okay, and of course they evolve starting in a particular place, so they have a common distribution. So these are all criteria that we can use to recognize a species. So now I'm going to start to give you two examples, two sort of case studies of my work with my students. And this is what got us started. This one in the middle here on the right, you see a fennel-leaved Lomaceum, or fennel-leaved biscuit root. On the left, you see what's called a Nevada biscuit root, courtesy of Jerry Carr's wonderful photography. And in the middle, you can see my attempt at photography we found a whole lot of things that look like this, which if we read one flora, the Intermountain flora, it would put this in with that. We said, when you collect several of these from several explorations of several populations, you say these populations look alike. I have a whole lot of things that look like this, but these don't quite look like populations of this thing. And they don't quite look like populations of that thing. And that's what I mean when I say they don't read the book, because I can't fit them anywhere. And why? Well, because they probably are something different. So we went out, collected a lot of these things. Kim Carlson then, in her attempt to understand if these were similar or different, measured a whole lot of characteristics. That's sort of where we start. And she found that these, as a group, were somewhere in between these and these. And as a result, we didn't know, well, are they the same? Are they just a big species and they're just really variable? Or are these names, the Nevada and the fennel-leaved, are those really good species and we have something new? How do you make those decisions? Well, we struck up at that point, and I've had a very strong collaboration since with Jim Smith at Boise State University. And we have now progressed considerably in our study of this group. And what Kim found, working with Jim and me, was that if you look at the genetics, and I'm not going to spend too much time here, but you can see genetically the fennel-leaved biscuit roots are up here, the Nevada biscuit roots are down here, and our plant is right here. Well, it's not really related to either of those, so we probably have a new species. Well, what is it? Well, it turns out there's a plant in California called Lomatium ravenii, or raven's biscuit root, and it was actually most closely related to that. And so we described it as we described it as a variety of this. Most people who look at it now are going to say, no, it really is a different species. But how do you make those decisions? We didn't know at the time. But in our collections, we also found some of this, which was a look-alike to this. And we thought it was the same thing at first. And when we put those two specimens in with the rest of everything, it didn't come out up here. It came out different still from either the fennel-leaved biscuit root or the raven's biscuit root. And so finally what we did is Kim did yet another study and found out that this, which we call bentonite lomatium, is located in one place in the world, which is that hill of very white bentonite clay in Sucker Creek in actually in southeastern Oregon. And that's the only place where it grows in the world. The raven's biscuit root grows more widely, but this thing only grows in one place. And so we actually described this as a new species as well. And here, when you look at the two of them, this one that we described is similar to the one in California, and this thing that we described, which is endemic, narrowly constrained in its geographic distribution to just these white ash hills in Sucker Creek of far eastern Oregon, right near the Nevada border. They're different. Now you can see it here, but it's pretty hard. You have to actually look at a lot of other characters that we didn't even notice at first. And sure enough, when you start looking at hairs and little fine structures that you often have to use in an identification key to distinguish some species, we found that they were in fact different. Okay, turn the page. Let's talk about a second example. As a result of the work of Kim, another student, Emma George, and I did quite a bit of exploring in Owyhee country, and she said, well, I'd like to do a project. And so what she did is she looked at biscuit roots from all over the Northwest and constructed this big, what we call a phylogenetic tree, list of relationships. Don't worry, you don't have to memorize all this. The point is, she focused on, she found one thing that we then moved on. So what I'm going to do is, as a result of her work, I'm going to go back to that collection that Meriwether Lewis made on the 6th of May in 1806. He was going back up the Clearwater River. This is the Clearwater River, and they're headed back up over Lolo Pass upriver. They came down, they passed this point on October 9th, 1805, on their way to the Pacific. But on their way back, they passed this point, and up somewhere in the confluence of the Potlatch and Clearwater Rivers, Meriwether Lewis collected that plant that we call nine-leaf biscuit root. And what I want to mention to you now is when we describe a species, we have to make reference to what we call the type, which is the population on which this abstraction, a species, is based. And so the name of the species, the nine-leaf biscuit root, is based on this specimen, which is housed in a herbarium in Philadelphia that was collected by Meriwether Lewis on the 6th of May, 1806. And if you look carefully, you can see that's a pretty wimpy thing. That's not a whole plant. That's a few leaves. You can kind of make out that it might be a nine-leaf biscuit root. You have no fruits, no roots. Well, it turns out we do have that population of plants that's still there. It was there in 1940. Lincoln Constance, a botanist out of Berkeley, went back to that same location. I figured this was his guess. I never asked Lincoln Constance about this, but his description of where he was is very similar to where Lewis was. And then I went back in 2016 and collected a plant that has the fruits and the root and the leaves, just like Lincoln Constance did. And so we kind of use this now as our base because it is what we call an isotype of this. It comes from the same place as Lewis's. And what we realized was all these nine-leaf biscuit roots that are all over the Pacific Northwest and actually look quite different. And you can just look at their fruits, which each species that came out in a different place in our genetic tree that shows relationship and ancestry. Each one of these rows is a different location in the tree, and you can see that they're a little bit different from one another, aren't they? Now, you all can see that at arm's length. That's pretty easy. But it's not so easy as it sounds. Well, we decided to, again, with another student, to look at this top row. You see where it says SPNOV 1, 2, 3, 4. Those are what we called species novum, which is new species that have not been identified that are thrown together because when you collect a plant, you put it somewhere in a herbarium and you throw it in a cabinet, and all of these things were thrown in cabinets that were called nine-leaf biscuit root, Lomatium tritornatum. And so we said, well, let's start to sort this out a little bit. And we took that first row there and we said, let's look at that. And so we collected a lot of that. This is what we called for a long time species novum 1, and this is in the Boise foothills. And that looks like this when you actually can see it and not in the background. This is what it looks like. And it looks a little like that thing I showed you a pressed specimen of. I'll show you some more in a minute. Here it is. That's the one that is in the Boise foothills. And there's the one up in by Lewiston, where the Potlatch River comes into the Clearwater River. They're a bit different. Are they different enough? I don't know. How do you figure it out? Well, you have to look at more. And so what we did is we said ecologically, they're very different. This is where the species novum 1, the one in the Boise foothills, grows in clays, and that one up in Lewiston grows in different soils and so forth. And genetically, the yellow or the red, which is what we're looking at in the Boise foothills, is a little different than the yellow and the green. And they look a little different, the red from the yellow and the green. And that's probably because they're different species. And sure enough, when you look at their genetics, the one that's red is different than the one that are yellow and green. We later resolved some yellow and green stuff, but that's another story. But look at here, this thing is different. So it's different morphologically. In other words, you can look at it and see that this is different than that, and this is different than that. And genetically, they're different. So we have a new species. We named this one after Governor Cecil Andrus, former governor of the state of Idaho, in honor of his work and his great contributions to wilderness and conservation in not only Idaho, but nationwide, as the Secretary of the Interior. So who cares? Why do we look at hairs, fruit shape, leaf form, DNA sequence, angels on the head of a pin, perhaps, huh? Why do we do this? Well, it turns out that this biscuit root group, as I mentioned earlier, the reason that Lewis collected several of these things is because the Nez Perce showed it to him and said, here, this is an important plant. Collected it. Later it was named. But lomatium is used by a lot of Native American cultures. Some as medicine, some as food, some as fiber. And a lot of them, like the bentonite biscuit root down in southern Idaho that we identified, are very, very narrow in their distribution. They're endemics, meaning they're localized only in a small geographic area. And when we talk about endemics, we have narrow endemics like the bentonite biscuit root, and we have broader endemics. And sagebrush, for example, is endemic to Western North America, but it's all over the place. That's a pretty broad distribution. But many lomatium, about half of the group of biscuit roots, are located in just very small places. And some of them are used by some Native Americans as food. Some of them are used by others medicinally. Some are used by others for other reasons. So species boundaries matter. And I'm gonna liken it to this. If you go to the medicine cabinet and you pull down a bottle of medicine and you want something to cure your headache, you look at the label and the label says acetaminophen, right? Or maybe you want Tylenol over here. And you pull it down and you take it and you trust that the label tells you what it is. Well, if you're not clear on your labeling, you might end up with a laxative, which would give you very different results than a painkiller. And as a result, it's really important to get our names associated with the right kind of plants, because the plant's chemistry's different in one place from another. Different species are adapted not only in looking different, but chemically different. And so the medicinal values may be different, and so on and so on. So all of this is made possible as a result of botanical exploration. New species emerged as a result of finding things that didn't read the book and exploring more in the laboratory. But you got to start where they live in the field. So what we do is we bring plants in from the field, we mount them like Wendy Harvey's doing down here on the bottom right, put a label on them that tells us where they were collected, when they were collected, where it was, and so on. And then we store them in these cabinets, and there they live. And we store them until we update them. So we've gone back and put Lomatium andrewsianum on the things that used to be called 9-leaf biscuit root, and so on and so on. And it's very important to have these things we call vouchers, which are examples of things that people saw in the field. And it takes a lot of time to collect them and bring them back. But it's very important to collect them and bring them back. And the reason is because, as Peter Raven, who's a very important botanist worldwide, out of Missouri Botanical Garden, the director of the Missouri Botanical Garden, a report without a voucher is only a rumor. How do you verify what that plant is that was really pretty up on a trail in sawtooths? You gotta know where it was, what it was grown with, what it looked like. Yeah, you don't have to genetically blueprint it, but you have to have a voucher in a herbarium to do anything to really understand it better than just take a picture of it. And so over the years, Dr. Packard, my predecessor, and I have built up the College of Idaho Herbarium to about 60,000 plants, and a lot of plants that are unique and nowhere else in the world, or that are type, what we call type specimens, are there, and at Boise State University. And so Jim Smith and I were able to get a grant to actually develop a resource that enables us, building on the heels of Ben Legler and David Giblin out of University of Washington. Ben is now, thankfully, in Idaho, at University of Idaho, working here, developed this database system. And Jim Smith and I got a grant, and I'm going to show you what we can do with this. I think I'm going to show you what we can do with this. Yeah, here we go. It worked in our prelim session. We have now about 60,000 specimens from Boise State University, about 60,000 specimens from College of Idaho, along with many other herbaria throughout the Pacific Northwest, totaling 3,080,000 specimens that are visible online. I'm going to show you how you can do this. If you search the database, we can go here and we can enter Lomatium, and I'm doing this as we speak, Lomatium andrewsianum. Whoops, it helps if you spell it right. And then we go up, I've got to move this out of the way so I can search, there we go. And what you get is a list of all of the plants, and I'm scrolling down on the right here. These are all specimens. You can see many of them are imaged because a lot of these are at Boise State University and the College of Idaho, because we really looked a lot to be able to do our work to identify that species. And there they are. That's where they come from. They're endemic to Idaho. They're a new species in Idaho. They're named after a prominent Idaho governor, Secretary of the Interior. And there you can see, and we can zoom in and we can see, oh, wow, well, they're not just in the Boise foothills, are they? They go all the way over here to near you. So keep your eye out for Lomatium andrewsianum, this fruit in your area. Okay, so let me go back now to where we were. That resource is available to anybody. All you have to do is go to this website, and there you are. Mary, is that where you should give me a thumbs up? Is that where you found these specimens? Yeah, yeah. Okay, so there it is. Welcome to explore there. This has changed the botany of Southern Idaho. 60,000 collections from art herbaria that are focused in Southern Idaho at College of Idaho and Boise State University. Now, those 60,000 specimens can be seen by anybody and are seen by anybody. And people who are studying these groups say, hey, we want to know more about that. Can you send us some of that stuff for DNA analysis? Hey, we want to know about that. And people are revising their conceptions of species. We go back and verify and we revisit. And this has been an incredible boost to understanding rare plant biology in Idaho and Idaho endemism. And here, what I've shown you here is an example of a map. This is a map of just one family of plants in this area that I told you that I'm studying. I just drew a little polygon. You can draw a polygon and say, okay, find all of this kind of plant in this area, and this is what comes up. And I can say, oh, look, boy, we haven't done much down there. Carol, you're in the audience. You know how we've been talking about going down to collect plants in there. That's because we don't know much about that area. So look at how it can guide further exploration. And that's a real boon to our work. So I'm going to shift gears now. And for the rest of my talk, I'm going to talk about two things. I'm going to talk about endemism. And the next several slides, you'll recognize Michael, who's in the audience. Most of the next set of slides are courtesy of Mike Mancuso, who gave a similar talk quite a while ago. And what I'm going to do is I'm going to look at the endemic plants that are found only in Idaho. And there are about 60. What is it exactly? Michael knows the number. But we've added some, like Andrus's biscuit root down here. I'm going to focus in this area where you are and talk about plants in the Wood River Valley, in the mountains of central Idaho, in the mountains of east central Idaho, in the mountains of west central Idaho. In other words, areas around where you are. And I'm going to talk about plants found only in Idaho. And I'm going to talk first about lower elevation plants, and then I'm going to move up to the tops of the mountains, because there are plants that are endemic to these regions of central Idaho at all different elevations, and you'll get the sense of that. And then after that, I'm going to talk a little bit about rare plants. And many of these, like this one, this is the Craters of the Moon cushion buckwheat, is locally endemic, but is not rare at all. In fact, there it is. You go to the Craters of the Moon, the cinders, and what do you see? This plant. That plant, Craters of the Moon cushion buckwheat. Now, Craters of the Moon cushion buckwheat is a slightly different variety than the other cushion buckwheats. Cushion buckwheats are all over the place. But this one, Eriogonum ovalifolium focarium, is only there, but it's all over the place. It's not rare at all. And that's where you find it. It's endemic to craters of the moon. That's it. So there are some endemic plants of the sagebrush steppe in the northern Snake River Plain and foothills, and I'm going to go through several of those now and show you some of them. Thank you, Michael. Peekaboo milk vetch is endemic to the northern Snake River Plain, and you can see here its typical habitat where it grows in these small sandy edges of these small playas in the sagebrush steppe in this region right here. Here's another one that's in the sagebrush steppe, the bugleg goldenweed, member of the Asteraceae, the composite family. And it grows, like you can see here, at the edges of these microplayas or ephemeral wet areas. So the habitat is unique for this plant, and that's what drove its speciation in all probability. There is where it is, endemic to just that region between about Craters of the Moon and over at the west end of Camas Prairie. Another one, Boise milk vetch, Camas Prairie to the Boise Mountains. There it is, endemic to that region. Now, some of these are plants you might have seen. White Frasera montana, grows in these dry meadows and forest openings, like along here and back up in there. And another plant that grows along the foothills, like say in the foothills of the mountains north of Fairfield, Mertensia campanulata, that grows on up into the Boise Mountains and the Smokies and so forth. Now, most of you have probably seen bluebells, but unless you're in this region here, you're probably not seeing that endemic one to Idaho. There are a whole lot of very common bluebells in the sagebrush, the little shorter guys, and there's a lot of them that are in the mountain streams all throughout Idaho and all throughout the Pacific Northwest, like ciliate-leaved bluebells. And then there's this one here, which is really interesting. Just a couple collections of this, a few collections I was able to find. And this was collected, the type of this is up between the Snake River, the Sawtooth National Recreation Area headquarters, just north of you, and Galena Summit in rock outcrops. Now, this is a really common genus called Bochara, which are the rock cress, but this rock cress is different than any other one and is found only in that region up by Galena. Now, there have been other collections. I have not had time to check those out. There have been 3 or 4 collections made in the last few years. And Mary, you mentioned you got to confirm those things. If they're not identified properly, that doesn't extend the range of this species. We have to confirm that they are, in fact, this species before we're able to really know the distribution of this. But how do we do it? Because collections were made, they're not just reports. And so we can go to those vouchers and say, nope, sorry, we have Rolenciorum isn't widespread. It really is only along that region between the Boulders and the Smokies, just north of Ketchum. And we don't know yet. This is one of those ongoing processes. Okay, we've got another suite of endemic plants from the valleys along Rocky Scree and volcanic rocks and outcrops in the Lemhi's Lost Rivers and the Chalice area. A lot of these, the Lemhi milk vetch, beautiful. Michael Mancuso knows more about these than I do. That's where the Lemhi milk vetch is. The dwarf penstemon, which is shown here, beautiful little penstemon species. And that's its habitat in the gravels in the Lost River and Lemhi valleys. There it is. Chalice crazy weed. This is in the legume family. Oxytropis, as some people say, I call it Oxytropis. Bessie Ivar salmonensis, just along the Salmon River in the Chalice Volcanics. Malm Gulch, for example, you've been up to Malm Gulch, a lot of really cool plants up there. Ashy slopes, there it is, the Salmon River headwaters on Chalice Volcanics, as is this wavy leaf telepody. This is another mustard family, and there's its habitat. Again, ashy outcrops, this is decomposing shale, and Chalice Volcanics in this region here. Again, narrow endemics. There's another big mustard family. This is a bladder pod. Actually, it's like a bladder pod. It's twin pods, two big bladders. Those are its fruits that you see there. And there you see the typical little mustard flower with 4 petals and so on. And that grows also on these shaly outcrops. Again, Michael will be better able to show you. This is not one of it. This is a penstemon, or yeah, it's penstemon, and that's it right there on that shaly outcrop. And now here's one that's a little more recent. This is one that was collected. It had been collected a while ago, but we only had enough specimens to actually know the variation of it. It's a Jacob's ladder, and this species was only described by Jessica Irwin as recently as about 4 years ago, 3 or 4, I'm not sure exactly. It's in moist partial shade of canyons in the northern Lost Rivers up to Ellis and May. Beautiful little thing. You probably know the Jacob's ladder, but this one is endemic to that region of the northern Lost Rivers up into the Salmon River area. Let's move up in elevation. We'll talk about plants in the rocks or talus up at whitebark pine communities near timberline. This is a favorite haunt of Michael's, who took these pictures. Many of these pictures, white cloud milk vetch that you see here, a very prostrate version, not like the Lemhi milk vetch that stands way up, but again, a legume, a member of the pea family. And there's its habitat up in the mountains. And there's its distribution, the White Cloud Mountains. That's endemic to the White Cloud Mountains, nowhere else in the world. Now, the next 3 I'm going to show you have a little broader distribution, and they are endemic to a wider area in the western and central mountains of Idaho. Everman's pincushion, closely related to the dusty maiden that you get commonly down at lower elevations. This is on scree slopes. And this one's very common. You've probably seen this one. It's very common in the mountains, Smoky Mountains, and the region going up through the Sawtooth, up into the all the way up Salmon River Mountains, Penstemon montanus idahoensis. And another one that is, again, not as common as the penstemon, but Idaho douglasia, which grows again in these gravels at high elevations. This one right at the edge of snowbanks. You can see that pink hue here. That's the douglasia. And there's its distribution. And Carol, if I'd known you were coming, I would have put in Lewisia sacagawea, which is over here. It's not quite as close to Ketchum, so I didn't include that one, but there are many more. I'm just giving you a sampling here. And then I'm going to show you another group of endemics. And these, the next 4 or 5 species are all members of the same genus, Draba or whitlow grass. And these species have very narrow and endemic, they're very narrowly endemic, most of them. And these are all Idaho endemics within the same genus. So we have 5 or more Drabas that are on rocky screes and crevices from Stanley Basin right up to the tops of peaks. This is the Stanley whitlow grass, Draba trichocarpa, and that is on these hillsides right above Stanley. There's Stanley, there's Horstman and Williams Peaks and the Sawtooths. And there in the foreground is the Stanley whitlow grass. Globefruit Draba, Draba sphaerocarpa. And Michael, you'll like this tribute to your old pal Star up on top of its preferred habitat of globefruit draba, and Michael's preferred habitat too, I think. And silver draba in the Sawtooth and Boise Mountains. You see here another endemic draba. Now, the last two I don't have pictures of in situ out in the field, but this one is restricted just to limestones. Many of our endemics are limestone, just like several of our endemics are restricted just to those chalice volcanic outcrops. But this and the next one, well, this one, the next one's a little broader ecological tolerance. Draba hitchcockii is restricted to limestones of the southern Lost and Lemhi Mountains. And the limestone draba actually is not restricted to limestone. There are collections of this off the limestone. Well, if you trust the labels, again, sometimes the labels say one rock type, and if you go into the field and they've got both rock types, sometimes they're not always growing in exactly the place that you think they're going to be growing. So that one's limestone draba. Okay, so now I'm going to shift gears, and for the last segment of the talk, I'm going to be talking about the rare plants of central Idaho. And again, we're going to move up in elevation. And I want to talk, these now are rare in Idaho, but generally are not always, but often more widespread throughout the Pacific Northwest. But they're really interesting things here in Idaho. We don't have many of a lot of these. For example, this one, this is the meadow pussy toes. And it happens to grow in wetter, saturated open meadows that get the hummocks in them. And you might have seen these in the valleys. There's a lot of them over in Birch Creek Valley and so on. These hummocks, and this is the places that are preferred grazing habitat. And so there are threats to these rare plants. And if you desire to become involved with the study of rare plants in Idaho, you can get much more information about this by coming to the Idaho Native Plant Society Rare Plant Working Groups, and I'll talk about that at the very end here. There's another really unique kind of habitat we call the fens, or the peatlands of the Stanley Basin, where you have wetlands that are not fed by streams, but rather by groundwater, and you get peat moss that grow here. And where you see that, you also get a lot of rare plants, including a lot of sedges, like you see here, the livid sedge, and like you see on the bottom right there, the brown bog sedge. But you also see some really oddballs, like this sundew. Many of you heard of these sundews? These are little guys that have little droplets that insects get caught in, little tiny flies, mosquito-sized crane flies and things like that. And then the leaves excrete digestive enzymes and basically digest the proteins so that these leaves can absorb the nitrogen from the insect that it's digested. And these insectivorous plants grow in often in nutrient depleted areas like these fens of central Idaho peatlands, Stanley Basin peatlands. Here's another one, these little bladderworts. These bladders are modified leaves. There you can see right there and there the flowers that come up out of the water, but the leaves are under the water, and the leaves have little modified sacs in them. And what the sacs do is you have a little hair on the end of the sac, and if an insect triggers that hair, that causes the sac to go whoop, like that, which creates a vacuum inside, an increased volume, which sucks water in, and with that water comes the insect. They close up and they digest the insect. So this is another insectivorous plant, Utricularia gibba. Very rare. You can see it only up in those Stanley Basin peatlands and some other ones up further north in Idaho. And there's a picture of its habitat and its what it looks like when you walk upon it up in above Alturas Lake, for example. Okay, and these are some of my favorites. If you move up to the higher wet meadows and subalpine seeps, this summer, Beth Corbin and I were up in this area here, and we saw more of these two species of Patricium than had previously been seen in Idaho. And right there up in the upper Wild Horse Basin, there's no trail up here. You have to kind of follow, well, there's a little old way trail, I think called Tilly Lake. When you get up above the lake into these seeps and subalpine meadows, you start seeing all kinds of things. Here's another moonwort that we collected up there that's slightly different. Look at the shape of those. Look at those leaves compared to those leaves. You see how separate the leaflets are? Those are two different species of the same genus, both moonworts. And these are ferns that have their spores released on separate leaflets than the leaf like you see here. These also, you're not going to find these unless you're down on your hands and knees. And that's how we saw those grape ferns. We're looking for plants on our hands and knees in those subalpine meadows. And here they are. This is another one, the Dane's dwarf gentian, chromosoma, and that's also there. Now here's one. How many of you have seen these? You've probably seen the grass of Parnassus in the stream sides and wetlands. It's a very common member of the saxifrage family. Well, yeah, something like a saxifrage. You've got these little fringed petals, beautiful little flower. Well, there's a diminutive, those things are about, I don't know, maybe a foot tall. These guys over here on the right are in the same genus, but are only about 3 inches tall. And they're only in these wet meadows, and there aren't very many populations of them known in Idaho. Is that because there aren't many in Idaho? Or is that because we just don't know how to find them and we haven't explored enough? Similarly, here's another one. This is the dwarf buttercup. Now that doesn't grow in these seeps. That grows way up in these rocks. And we only know of this from a couple of populations up by Cane Lake, in the cirque above Cane Lake, which is in the Pioneers. So here I'm in another cirque. We're looking for plants. We never found this one there, but I don't think we were in quite the right habitat because we got so sidetracked getting these other little meadow plants that we never saw these. But they're up there to be found. We only know of them in Cane Lake. Is that the only place they are? Further botanical exploration will tell. Then there's a lot of what we call sedges. They look like grasses. They're a little different, that grow in alpine and subalpine wet areas like you see here. This is one of them. There's another one here, which is called the fetid sedge or native sedge. They grow in talus slopes where there's some sub-irrigated water. They're moist sites, but they're gravelly. They're not really meadows per se. And then there are several others that I'm not showing you because I want to get through and show you a few. This is one that this tells a very interesting story. This plant, which is called Hall's Rush. Again, Beth and Michael and I were up in a different cirque, Fall Creek, and exploring habitats like this. We came across this plant that's in the middle here, and we figured out, okay, it's Hall's Rush. Well, is that rare in Idaho? And we saw, oh, yeah, it's pretty rare, but there are probably a dozen collections of it. Well, it turned out that every one of those other collections was something other than Hall's Rush. So there's only one known population of Hall's Rush. You're looking at a plant that was collected from that population. That's what it looks like. It's more common in other states, but we know it of it in one place in Idaho.
Don,
Go ahead.
we have a question.
Yes, go.
If you could go back to grass of Parnassus, please.
Sure. Whoops.
So the question is from C. Norton, and it says, I've heard the term grass in quotes used, such as grass of Parnassus for flowering plants. Is there a specific reason for using the term grass? I would not have expected to look for a flowering plant called a grass.
Yeah, there's a lot of that going around. There are lots of plants that are called mosses, for example. The hanging moss, the Spanish moss. Well, that's in the pineapple family, which is a flowering plant that does have showy petals. Okay, well, showy, they're small, but grasses don't have showy petals, unlike this. No, there isn't. I don't think there's rhyme or reason to that. But there are lots of examples of that. Yeah, so I don't know if I've answered the question, but I mean, you look at the leaves of this one I just showed you the flower because you've probably seen it before, but you look at these leaves, they're very broad, they're not grass-like at all, and that's not what a grass flower looks like. Grass flowers don't have petals. Well, they have them, but they're reduced. They're only microscopic. You never see them unless you're looking under a very high magnification of microscope. Any others? I'm sorry, Lisa, I was going to stop more along the way. If people have questions, I'll entertain them. I'm almost done here. And I've just got a couple more slides and then we're going to come to a close. Another group, you probably know cinquefoils. Probably many of you probably grow the shrubby cinquefoil in your yard if you would like to grow natives. But this is one that's up high elevation. It's not terribly rare in Idaho, but there are several. This is what this flower looks like up close. There are several cinquefoils that are rare in Idaho, and we don't even know of them. Barbara Erder, who's a cinquefoil fanatic, wrote The Flora of North America treatment of this particular group of plants. And there are probably, I think she has 2 or 3 or 4 species that we don't know anything about because they're way up high on these ridges and haven't been collected. Other plants that are way up high on the ridges, like this was only described how long ago, 20 years ago maybe, and it grows up. This one, probably Michael took that up on Sheep Mountain, I would guess. And then I took the lousier shot down here on the bottom. Up here above Horseheaven Pass, there's Bora. It's at high peaks up in the Lost Rivers and the Lemhis. And I'm going to end, I think this is the lot. Yeah, I'm going to end my talk on this because this plant is, if you live if you were to live in Ketchum, some of you are probably in Ketchum, this is probably the closest rare plant to you. It's up on the ridge of the Boulder Mountains. There's the Boulder Mountain Ridge over there. If you go to Lake Creek or Basin Creek on the Trail Creek side, Lake Creek on the Big Woods side, and right in between is the high ridge of the Pioneers. And at 10,300 feet, up right in rocks, right on top of the ridge is this. This is the only collection of this genus or species in the state of Idaho. The closest collection to the ones up on the Boulder Mountains collected by oh goodness, Bob Mosley in 1987. This is the only population. He said it was very common up there, and I'm hoping to get back up and see if I can find more of that and other populations. But that's probably the closest one to you. And again, the closest plant in this species to this one on the Boulder Mountains is in the Ruby Mountains in Nevada. And the rest of it's down in Nevada, Nye County, which as the name implies, Nye County Falls, Candy Tuft. The last, now I'm done with my flower slides, but I want to mention that explorations are ongoing. And I want to just mention that you are invited to join in botanical forays. We've had 13 of them for 13 consecutive years, including last year where we all were masked up over in the Caribou Mountains over here, 13. This summer, we're going to go up east of the Salmon, east of the town of Salmon, up in the Bitterroot Range, gather at Cougar Point Campground from the 15th to 18th of July. If you want to hang out with some botanists and go explore, join us. You're welcome to join us. To give you an example of some of the things that we find, here in the middle, these are 3 different kinds of cinquefoils. The one on the right, and they look different, don't they? Of course they do. They're different species. The one on the right is a well-known species called slender cinquefoil. The one in the middle is blue leaf cinquefoil. And the one on the left is an undescribed species that's obviously different. It grows here. This is it, out in Ayers Meadow up in Bear Valley. We found it in 2017. Haven't got around to describing it. Trying to get some money to raise to do it properly. At any rate, we're going to probably have a well, I don't know. We were able to fund our work on Lomatium andrewsianum by having a crowdfunding opportunity. Also, if you're interested in the rare plant piece, we have 2 working group meetings a year. And in February of 2022, every 2 years we have a conference. We made it just before the lockdown last February, just before everything closed up. And so our next rare plant conference will be February 2022. And you can find that out by looking at the Idaho Native Plant Society website. It'll be announced. So that's all I have. And if there are any questions, I'll entertain them as long as you can stay.
Okay, so I have a question, and I want to, anyone else who has questions, please feel free to type them in the chat box, so we're not all talking on top of each other. So here's my question. I took plant taxonomy back in the end of the 1970s, a really long time ago, and we didn't use things like phylogenetic trees and charts. So would you talk about that a little bit so some of the more lay people in the audience can get an idea what you're talking about?
When I talk about, let me pull up an example here. I think this was the first one here. What we did to generate this picture is we took DNA that we extracted from fresh tissues of plants that we collected in the field, the real thing. We took DNA and we extracted the DNA. And as you know, DNA consists of a sequence of base pairs. So we found, I think we ended up with 4 different genes, some from the nucleus and some from the chloroplast. And we sequenced the bases in that DNA and ended up with about, I think in this study, we probably only had about 1,500 or 2,000 bases. Studies we're doing now, we have on the order of 40 or 50,000 bases. And what you can do is you can line them up, and one will say, you know, there's 4 letters in the genetic code, right? A, T, C, and G. Okay. So if you have, imagine 1,000 letters in a row. So A, T, T, G, G, C, C, A, T, C, G, okay, and so on. You get the idea. And then another specimen has, instead of A, T, T, G, G, C, C, it says A, T, T, G, 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 C. It's a little different genetically, and so on. And based on, it's a little more complex than just similarity, but I think if you just get that, the more bases you have in common with another species or another population, the more likely you are to be closely related, because there have been fewer changes in your DNA, the more similar you are. The more different you are, then that means that it's been a while since you emerged from a common ancestor. So what you have here is a tree. We call it a phylogenetic tree. You can see it as genetic relationships. So all of these right here, which are all samples of DNA that came from plants that look like this. Okay, that's the arrow, the circle, Raven's biscuit root, this one. All are similar enough to one another that you can't really separate them much. And these numbers just tell how reliable our picture is. And these things are more closely related to the plants in California that have the same name. That is, their DNA is more similar to the DNA sequence in those base pairs from those plants than they are from these plants, which are way far away genetically. Is that a satisfactory answer, Lisa? Or you want to pursue that?
Absolutely. So there's a follow-up question.
Yes.
Are individual plants as genetically different as humans? Meaning there's something like a 1 in 6 billion chance now that someone else matches your exact DNA. Is the same true for plants? And
I don't know. I don't know if anybody's done that precise kind of comparison, but I would guess that certainly individuals within a population would likely have that level of variation. You know, that small amount of variation. But between species, the further you get, even if you call it the same species, like here, you know, these two, these all have the same name. These are all from California, and they all are what we call Lomatium irvinii. These two are more similar to one another than this one is. But how many bases does that differ? I'd have to go back and count and look at the data. But we have the data. I can do that for you. But I've never really bothered to take the time to do that, to be honest with you. That's a good question, though. Did that answer it?
The comment says, neat, and then it says, I've heard of crimes being solved based on plant evidence and specific locations.
Oh, yes. You could take a piece of DNA and run it through a sequence it, and see who it comes out with here, and you'll say it's this species. And if that species doesn't grow at the scene of a crime, well, I don't know. I don't know where I can't picture the whole context. But the point is, if you get a piece of DNA from that plant at a scene of a crime, then you know that plant was growing there, right? So that, yeah, you can do the same thing, like the forensic botany of your generation and mine. When we read about the forensic botany, it wasn't DNA sequence; it was matching the growth rings in the tree that was used to kidnap Lindbergh's kid to the trees that were growing in the backyard of the guy who made the ladder that kidnapped her. Yeah. And so, yes, there's forensic botany and there's forensic botany, and it still happens.
Yes. Pollen grains, for example.
What's that?
Pollen grains.
Yes, pollen grains. Yeah, there's a lot of morphological variation in plants, and each bit of it, whether it's genetic variation in DNA sequences, or whether it's pollen grains or wood grains or hairs on the leaf of a biscuit root, it's informative.
So does anyone else have another question? If so, please put it in the chat. Well, splitting hairs, it says. Thank you.
Yes.
Thank you so much, Dr. Mansfield. It was a very interesting talk. I'm going to stop the recording now. If anyone still has a question,
before you stop the recording, if you want to follow up anything, don't hesitate to email me, dmansfield@collegeofidaho.edu, and I will respond.
Fantastic. Thank you so much, and thank you all to our audience for coming to our Wood River chapter's presentation tonight.